Good morning, good afternoon, good evening, wherever you are. And thank you for assisting today at this fireside chat, drug treatments and therapies for dementia. It is a first of a new series that we are doing. And we are going to do it quite quickly. So whilst everybody's logging in, I'm going to go through some practical topics. Next slide, please. I am Paola Barbarino. I'm the CEO of Alzheimer's Disease International. ADI was established in 1994. It's the umbrella organization of 105 Alzheimer's and dementia associations around the world. We represent you all at the WHO, the UN, the OECD, and other global and regional bodies. We are the promoter of World Alzheimer's Month and the publisher of the World Alzheimer's Report and a lot of other reports. Our vision is to prevent, care, and include today, whilst we are waiting for a cure tomorrow, which is the topic of this chat today. Next slide, please. Just to remind you that these chats are free, and if anybody would like to make a donation, we would be terribly grateful, and thanking the donors that have given to us in the past. Next slide, please. So I have introduced myself already. Let me now introduce our star speaker of the day, Professor Alireza Atri, who is the medical doctor and is at the Banner Sun Health Research Institute, but critically is one of the foremost researchers in Alzheimer and dementia. He's involved in a lot of the new therapies that are coming out. We also have the honor of having him as our board member and the chair of our medical and scientific panel. Next slide, please. So this is a series of new discussions. It's going to be about 30 minutes in length, so we're going to take it quite quickly. We've been receiving a lot of questions, and I can let you know that we are not going to answer questions individually. We've grouped them in broad groups, and I'm going to ask those questions to Ali in these broad groups, and hoping that a lot of your questions are going to be addressed. Some of the very specific ones, unfortunately, we won't be able to. You can introduce yourself in the chat, as some of you are already doing, and you can also pose new questions in the Q&A. Should we have more time, we will make sure to address those. And with that, next slide, please. I actually welcome Professor Atri, and I will ask Ali the first question. So, Ali, welcome to the fireside chat. It's a pleasure to have you with us. It's very early in the morning for you, so thank you for being with us. How early in the morning is it?
It's early. It's still dark. It's quite dark still.
So thank you for making the time. The first question for you is around monoclonal antibodies, the famous mAbs. So aducanumab, lecanemab, we have heard a lot about these mAbs, but many people on this chat will not know what mAbs actually are. What are they treating? What are they addressing? Tell us a bit more about that.
Yeah, monoclonal antibodies are been around for a while, and they're being more used in many different types of illnesses and diseases. It's a way of helping the immune system very quickly target and attack something that we want to get rid of. So basically, they're proteins that are made by our body naturally as part of the immune system. And they target what we call antigens, something that our body tags as being foreign. And once that gets tagged, then other parts of the immune system, if you think about it almost like having an army, and you have scouts, and you have the Marines, and you have the Army and the Navy and the different. These are sort of scouts that tag things so that other parts of the army of defenders can get rid of them. And in COVID, for example, they made mAbs, and they usually give it to you by either an infusion or a shot, and it gives you immediately, your body can target something. They've been used in immune disorders, cancers. So a lot of the progress that we have, for example, with some of the cancers is identifying some part of a cancer cell, making a protein against it, and then helping our own immune system get rid of it. They can be made in different ways. They can be made from getting blood from people. So aducanumab was actually one where they looked for these antibodies to amyloid protein, which is a major constituent of Alzheimer's. And they took that, they purified it, and made many of them. So they call monoclonal. So one kind, and then they clone it. They make more of it. So that was a human kind. You can also make it, using basically animal part, using antibodies from animals, or sticking them together with human-derived antibodies. So it's a very quick way of targeting something and using the immune system to get rid of it. And we've been studying the monoclonal antibodies for a long time in Alzheimer's disease, over 20 years. And initially, interesting enough, you mentioned vaccines. Initially, our first thoughts were, let's do vaccines. Then things went off the rails with the vaccines, and they said, well, maybe we can do the monoclonals first. And that's where you've had multiple generations of monoclonals, mostly targeted to amyloid protein. And now we're going to try to target it to other things like the tangles, the tau. But, progress takes a decade or more, sometimes 2 decades in this case.
Indeed, and it is taking a lot of time. And I know that a lot of people online will be wondering when things are coming up. Mm-hmm. So you've talked to us about how monoclonal antibodies work. And you've mentioned vaccines. So a lot of questions that we have received are around that. Quite a lot of people don't quite understand the mechanism of vaccines either. So we are used to vaccine targeting something that is contagious, but of course we are talking here of a non-communicable disease. So how do the vaccine work? What kind of vaccine are out there that may be promising? I mean, or at least what kind of timing are we looking at for something like that?
Everything's promising. These are approaches. So they don't tell us, there are ways of us attacking some part of what goes wrong in Alzheimer's. So again, I think one of the main things that will accelerate all our treatments and pharmacological treatments for Alzheimer's is the fact that we have biomarkers now. So we can finally measure in a living person the plaques, the tangles, and we don't have to have brain tissue for that. So the initial part, and it's again taken us 20-something years to do this, is to get spinal fluid. So in the spinal fluid that bathes the brain, you can find these proteins. So that's one way, but that requires some expertise. And then we have the PET scans, but the PET scans are expensive. And when you do a PET scan, you only do it for one thing at a time. So you look at amyloid plaques, or you look at tangles. Now we are working on blood biomarkers. Some are available, but not validated. So in the next 3, 4, 5 years, look for more validated ways of taking blood and being able to tell whether you probably have high levels of plaque or tangles in the brain. The other parts of the system that we look at is the effect of what happens when the fire starts in the brain, which is inflammation. There's good inflammation, there's bad inflammation. And so we can probably find biomarkers for inflammation we're working on, and for destruction of synapses. This is a roundabout way of coming back to saying that vaccines are an approach that were used early, and they used a different part of the immune system. So if you give a vaccine, you don't immediately tomorrow have protection. Actually, vaccines have to activate and produce things like antibodies and alert different parts of the immune system. And what happens is you have to oftentimes give a piece of a protein or something, whether it's a virus or a bacteria or a cancer cell, right? Or parts of a cancer cell, and hope or expect, by a lot of the studies that you've done in animals and things like that, that it gives you a big enough response that it recognizes it and activates the right parts of the immune system. So with the vaccine, it's more complicated. It's more complicated than the monoclonal. And the initial vaccine was done, and what it did is it caused about 6% of the individuals who received it, this was 20-something years ago, to have too much inflammation in the brain and encephalitis. Was it able to clear some of the plaques? It was able to clear some of the plaques, but it was too much. It activated what we call T cells. So for quite a while, they thought that meant we didn't know enough, so we went the monoclonal way. Actually, vaccine research is alive. There are a number of vaccines that are being tested. One of my affiliations is with Brigham Women's Hospital and Harvard Medical School. I'm not part of this vaccine trial, but they have a intranasal vaccine that they're testing. There's a Swiss company that's been doing this for a long time, and they're involved with it. There's been a vaccine that they've Down syndrome, individuals with Down syndrome also have plaques and tangles when they get older. So it's been tested in that population. The issue is, can you get the right parts of the immune system activated with a vaccine? That's been a problem with a lot of them, or not causing the wrong part to get activated too much. But that's absolutely an approach that in the future, we will have more of, and we think that it's something that probably in midlife for prevention would be very, very important, but we still have a lot to learn.
Well, I mean, that is fascinating, and thank you also for reminding us that this is a life course disease, and therefore there are issues around it at any moment in the life, and we're still trying to understand at what stage, not just of the disease, but of the life of the individual, every treatment may give the best outcome. And this is in part why it's so complex, as well as the disease itself being so complex and manifesting itself in so many ways. Now, very briefly, I'd like to remind those online, I can see we are 221 now, that the chat is for introducing yourself. The Q&A is for questions if you have them. And if we got some time, we may try and pick up a few of them. The questions I'm asking are the one that a lot of people have sent in advance and that we have put together in a few overall questions. I can also remind you there is an AI translation available, and in the chat line there are details how you can have your automatic translation on. Now, Ali, the next question is something that a lot of our members of ADIs particularly, are very keen on and really would like to know more of, and that is around the role of supplements, of vitamins. Everybody talks about these things. Recently, one of our colleagues, Chris, came back from America, and he said that he had this wonderful phrase, you cannot blackberry your way out of this, or blueberry your way out of this. And that's a classic thing, people fell ill and then somebody will tell you there is this or that you could have taken to be better. But is there actually any evidence of any of these things working?
In a simple answer, no. Working meaning there's just not the level of evidence like there is with, obviously the approved pharmacological treatment. And let's not forget, there are approved treatments that have been around for a while and are generic in most countries. There isn't that level. And I've been involved in so many of them. The issue is at the stage where somebody already has Alzheimer's dementia, and they've had the disease, Alzheimer's disease, progressing usually 25 or 30 years in the brain, from forming different toxic forms of amyloid to them becoming plaques, getting deposited in different parts of the brain, activating neuroinflammation, then causing tangles, or tangles appearing, and Alzheimer's kind of fanning the flame, kind of pouring gasoline on the flame. And now the tangles go to different places. And now you have vascular damage, etc. It's, eating blueberries may have some antioxidant properties, which is great. But that by itself probably is going to be something that can moderate and slow down something for some people to some point, but not enough for us to detect in big studies. And you hear the same thing about coconut oil, and you hear it about omega-3 fatty acids, and fish oils, and vitamin B12 and folic acid, and I can go on and on and on and on. And it could be that in midlife, that's something that was going to really help you a bit more, help against inflammation. But once you already have the, so much damage, our studies haven't shown it. There's only evidence in Alzheimer's disease dementia for vitamin E. And there are two studies. There's a Mary Sano et al. New England Journal of Medicine. You can look it up. It's the late 1990s, like 1997. And then there's a study again, I think 2014, Dysken, Dysken. And interesting enough, they showed exactly the same thing, that for some reason, and we don't understand again why, high dose vitamin E, that's 1,000 units basically twice a day. We need it twice a day for it to get into the brain to a certain amount, and you probably need about 800 units for it to actually get into the brain. Both of them showed that they slow down the progression of functional decline, not cognitive decline, functional decline by about 20% per year for the duration of those studies, which, they're not 5 or 10-year studies. So there's something about them, about maybe learned tasks or other things that they may help. But again, even vitamin E, you have to think about it, it's a little bit of a blood thinner. So people have had a lot of, little bleeds in their brain or on blood thinners. We don't give it to them. And it was interesting enough, we used to give it in the early 2000s. We could give it to everybody for the most part. And then there was a meta-analysis that came out. I think it was the Annals of Internal Medicine or something like that. And then it sort of suggested that, well, maybe vitamin E causes a higher mortality risk. And then people stopped giving it. And basically, it was about 4% more, and it wasn't in Alzheimer's patients. It basically took a meta-analysis of a bunch of other studies and put it all together and picked out the vitamin E and said, well, maybe it's not good for everybody, but you could actually see for folks from the Alzheimer's that it didn't cause any issues. So I still recommend it to patients. I would suggest eat a balanced diet, probably for prevention and slowing cognitive decline. The best evidence is for the MIND diet, which is the Mediterranean diet with the anti-hypertensive diet. So look it up, MIND, M-I-N-D diet. And there's probably the best evidence base for that. And there are studies that are ongoing. There are large studies around the world that are looking at the influence of diet combined with exercise, combined with social and mental engagement, combined with decreasing your vascular risk. They're going to tell us, hopefully, how much we can slow the process down. But I think people become very hopeful and they hear about curcumin and things like that. We've been involved with a whole bunch of these things, and some of these things don't even get into the brain. So I wish I could be that hopeful to say we're going to find that berry that's going to actually, reverse everything. It's a complex brain disease that has multiple components, and we're unlikely to treat it even with one drug. We're going to need probably multiple drugs at different stages.
Thank you very much, Ali. I'm sure a lot of the people asking questions, you really covered that. There was a lot of questions whilst you were speaking asked that you did cover. On the vitamin E, I think this is good news. A lot of people will find that useful, but it's important to remember again, not everything works for everybody. The dosage, the individual issues that may also make something counterindicated for that particular person. You cannot just say this is going to work for everybody.
That's right. Just because it's a nutraceutical doesn't mean it's safe. That it doesn't interact with your other medicines. That actually the quality of it is right. In the U.S., they're not regulated. So things that don't go under a GMP, good manufacturing practices, you can again look it up. Every few years, there are these studies, and they look at these supplements, and they find that half the ingredients aren't what they're supposed to be, or they have some other ingredients in them. So if you're going to do a supplement, make sure it doesn't interact with your medications, and make sure you get it from a good source that is GMP.
Yes, and also things change. So one of the questions was around Oligomanate, that of course did go around, and a lot of people were very hopeful about that. It was about seaweed. So what happened there?
Yeah, it's interesting because the idea was, well, it's derived from seaweed, so it's got to be not a drug. I mean, I think people don't appreciate that all our drugs come from a natural source. We just purify the active ingredients. So this was a drug that came out of China, and the studies were done there. And it was interesting because they were done not on the background of what we would consider standard of care. And standard of care, once you have dementia level, is to start a cholinesterase inhibitor, okay? And so that we already know gives people a boost. And it was not done on that background. And then it didn't show, a positivity in all their population. There was a bit of a slicing and dicing of the data. So we said, okay, great. So it was approved in China, but the studies had to be done outside China. And the studies were started in the US. And again, they were doing it for people who didn't want to be on the approved drugs. And out of nowhere, the study stopped. So we don't know, I don't know why. But again, it's one of those things that makes us skeptical sometimes when studies are done, but they're not done internationally or through I'll just say that once you get these results, they have to be replicated. That's the most important part. And they have to be done in a rigorous way so that we're making sure that it's double-blind, placebo, randomized, and ultimately that they have the effects that are both positive but also safe. So I don't know what happened to Oligomanate, but it's no longer, the studies are no longer going on in the US.
Thank you so much. This is all very helpful. And I hope, again, those that ask those questions are finding your answer very illuminating. One set of questions that we received was around the non-pharmacological interventions and the fact that they are equally important. And I think a lot of the people following us know full well that ADI and all the people around us, like Ali, are the first to say that that is the most important thing that you can do, and the first and foremost, and still the only thing that we can do that really makes a difference. So Ali, do you want to expand on that?
Yeah, I always, in my talks, I talk about kind of like the 4 pillars. Like, I'm sitting on a chair right now. We need, all parts of the chair, and we need all the legs, so to sit properly, or else we're going to fall off. I would say the very first part of the leg of the chair should be a timely, accurate, and a compassionate, patient-centered diagnosis. So if you don't diagnose people, right, you're denying care. It's denying care. It's denying autonomy. It's denying justice. The second part of it is education about what this means, and doing all the things like shoring up support systems and communication, and teaching the dyad, the patient and the care partners, that this is a journey, and over time there's going to be environments, communication, all those things have to change. And sometimes there's triggers of things, what to be careful of, what to be wary of. And then you get to pharmacological. And the first thing I do is to take away meds. People have been piled on meds because they appeared maybe sad or depressed, but really they had apathy, they had loss of withdrawal, they had sleep problems. All those things can also suppress cognitive function. So then you add the regular medicines, and then you really put this through the care partner, and you get this strong dyadic relationship with the care partner and the patient. So they're not mutually exclusive. At the same time, probably psychosocial interventions aren't going to, completely change everything. So for example, if you have agitation, aggression, and somebody who is screaming, shouting, scratching, punching, resisting care, won't let you, wash or bathe them. Now they're not taking medicines. There are many things that you can do and must do before resorting to, potentially medicines. and those are all psychosocial interventions. But there comes a time when they're just not enough. And that's why all of it is important, because we're not going to have a cure for these diseases for a while, even though we finally got a toehold with biomarkers. And as we kicked off the talk about MABs, we finally have MABs that are clearing amyloid, right? We're finally showing that we can clear amyloid from the brain. And by doing that, we're slowing decline by about 25 to 35%, okay? Over 18 months. These are 18-month studies. So that's worth about 6 months of time, extra time at the earlier stages for patients and families. If this goes on, we add other approaches, psychosocial interventions, and we can hopefully expand that time. So it is a really hopeful time, and psychosocial interventions are very, very important. And but neither drugs or them alone are gonna do the trick at this point.
Thank you very much, Ali. Some of you may have noticed that I've had to change position because my internet connection, I think, is not very stable today. So apologies for that. We are receiving a lot of questions. A lot of them, Ali, you have already tackled, but there are some questions around the use of blood biomarkers. You've mentioned them briefly at the beginning. Would you like to say a little bit more? Because these could be transformative.
Yeah, so it's transformative. It's going to be transformative. You're right. However, they're not validated in diverse populations. So the spinal fluid, we've been able to work on for 20-something years. The levels of the proteins for amyloid protein, for phospho-tau, different kinds of phospho-tau, for markers of inflammation, neurodegeneration that we're still developing. The gold standard really from them, for a biomarker is from the spinal fluid. And the levels are just so much higher because there's this thing called the blood-brain barrier that prevents things from the blood, for the most part, getting into the brain. Also, because of all the different other kinds of proteins and other things that circulate in the blood, these can be affected by your genetics, your racial background, your weight, your kidney function, your comorbidities, the medicines that you take. So while we're getting closer to the blood biomarkers, they're not, it's not like I can say for everybody, oh, this is 100% positive. There are these curves, and some parts you can be very, very confident that they're positive or high, some very confident that actually they're low, and then there's this gray zone. And we have to figure out what that is for everybody, and develop different markers. So there's amyloid markers, there are phospho-tau markers, there are other markers that are coming along. And ultimately, what's going to happen is probably we're going to use it for screening much more. And then in some people, we may need to, they may have the gray sort of shades in the middle. They may have to go on to do spinal fluid or a PET scan for confirmation. But it's going to change things. So imagine, again, let's just take the paradigm of our most advanced therapies right now, which is the monoclonals, right? So for the monoclonals, one of the things that prevented us from advancing it was, we didn't have markers early on. So about 20% of the people in the early studies didn't have enough amyloid in their brain. So now we can pick those people out. There were safety issues that we thought we may not be able to overcome, and in clinical trials we have. Now in the real world, there has to be guardrails around monoclonal antibodies. But second, imagine if you go into a study, they showed that you have a certain amount of plaques and you're getting plaque-lowering drugs, then maybe later on you can correlate that in the beginning, the PET scan with the blood, and then over time, follow the blood to show the levels have fallen. Just like when you take anti-cholesterol, the cholesterol-lowering medicines, right? So you measure your cholesterol, you give the medicine, and then you come back to see how much has fallen. And if we can have that for the blood, it's going to be easier. It's going to be more accessible. More people can have it. It'll be cheaper, and we may be able to also hopefully look at the aspects of safety and how much it's lowering, what effects it's going to have on therapies. So very hopeful time, for biomarkers. It's going to accelerate every single type of approach, whether it's a vaccine approach, monoclonal approach, whether you're targeting inflammation, tangles, amyloid at different stages, vascular, energetics. As you know, there are medicines that there's diabetes medicines, for example, that are and weight loss medicines that are being tried for Alzheimer's disease, not because they're lowering amyloid, because there's been a signal in their studies that it can potentially delay dementia. And the thought is that it's working on neuroinflammation and energetics.
And that is so exciting, isn't it? That is a really positive note on which to end our conversation. You've got to catch a plane, I know that. And for a lot of you that have been into this fireside chat, we'd like to have your feedback of what you thought about the shorter format. We're going to do more of them. And this year theme for us is going to be in World Alzheimer's Month, risk reduction. So we will come back to diet, we will come back to food quite a lot of time during the year. So expect a lot more detail. But today I just want to thank Ali for giving us the time and his immense brain and science to share with us all of these issues that are really difficult to explain in 30 minutes, but he has managed once again to do that. Grazie mille, Ali. Have a lovely safe sleep.
Bye, everybody. Thank you. Thank you. Thanks for all the work you do. Appreciate it. All right.
Grazie, Ali. Ciao, ciao. Thank you, everybody, for being on the line. There was over 230. I lost count at some point. Thank you for being with us, and looking forward to seeing you again at another chat or webinar soon. From all of us at ADI, thank you and goodbye.